Hey CW Bio, Mr. Kennedy here, and these are your lecture notes from Chapter 11 on meiosis. You might remember from a previous video that Mr. Kennedy taught you a whole lot about a type of cell division that sounds very similar to that word I just said. That other type of cell division is called mitosis. So this is meiosis. Notice there's no T. Don't get them confused. Now, why do we have two types of cell division? Well, just a quick recap on mitosis. If you recall from that previous video, mitosis is a type of cell division that is absolutely necessary for growth, maintenance, and repair processes in our body. And at the end of mitosis, basically we have a cell that is the exact same as the one we started with, okay? So we're going to use mitosis for skin cells to replace skin cells, muscle cells to replace muscle cells. You get the idea, right? And barring any kind of mistake in the division process, well, like I said, the cell that is produced from mitotic division is an exact copy of what you started with. That's what we want for growth, maintenance, and repair. There's one problem with mitotic division, especially in an organism like us. You can't use cells from mitosis for reproduction. In sexually reproducing organisms like humans and cats and dogs and fish and frogs and even things like plants, you need two cells to come together from two separate individuals. Going back to mitosis, in humans, if you have a cell with 46 chromosomes that divides and produces a cell with 46 chromosomes, you can't take a 46 chromosomed cell from one person and combine it with a 46 chromosomed cell from another person and end up with a baby that still only has 46 chromosomes, because I don't know, 46 plus 46 doesn't equal 46. It's 92. That's too many chromosomes. It doesn't work. So there you have it, ladies and gentlemen. That's why we need this type of cell division, called meiosis. Meiosis is all about taking those cells that normally, in a human anyway, would have 46 chromosomes and reducing the number by 1/2 for sexual reproduction. Let's look at how this works. And as I go through this with you, I'm going to draw lots of parallels and comparisons between meiosis and mitosis to help you keep them separate and to help you remember how both of them work. We're going to start with the idea of genes and chromosomes, okay? So if you remember from that past video in mitosis, I taught you that inside the nucleus of every cell, that's where our DNA lives, and it's organized into these X-like things called chromosomes. And those chromosomes carry our DNA that's passed on from one generation to the next. Well, I need to add a term to that, and that term is the word gene from this slide. So that DNA on those chromosomes that's passed from one generation to the next is actually organized into small bits of information called genes. Each gene is responsible for the production of a single protein, which translates up, as we'll learn in our unit on genetics, to a trait of some sort that you see in the mirror. Like you'll have a gene for your hair color, a gene for your eye color, genes for your skin color, you name it. There's genes for that, right? So those genes are basically chunks of DNA organized, right, one to the next on chromosomes that, again, are passed from one generation to the next from the nucleus of a cell, okay? Now, you may also remember that we talked about two terms in that other video, diploid and haploid, okay? Your body cells are considered diploid cells because they have both sets of homologous chromosomes that you've inherited from your parents, right? Remember, each parent in humans anyway is going to contribute 23 chromosomes through their sex cells to their offspring. So you have a matched set of chromosomes from mom and a matched set of chromosomes from dad that come together to make baby. That's how it works. Again, doesn't matter if we're talking about humans, cats, dogs, fish, frogs, or even plants. Anything that sexually reproduces, that's how it works. two separate cells from two separate individuals have matched sets of chromosomes that come together. Those matched sets we call homologous chromosomes. And when both of those sets are in a cell, that cell's a diploid cell. So that diploid number for a human would be 46. We have 46 chromosomes in every one of our body cells. And because there's 46 chromosomes in every one of our body cells, that also means we can't use those for reproduction. So why can't we use them for reproduction? Because if you remember at the beginning of this video, I said 46 plus 46 does not equal 46. To have a functional human, we need to have 46 chromosomes in our cells. If you took 46 chromosomes from a female and combined it with 46 chromosomes from a male, your offspring would have 92 chromosomes. That's 46 chromosomes too many. So there has to be a way to reduce the number of chromosomes in these cells to get a diploid cell down to haploid structure. To be a haploid cell, you have half of the chromosomes in the cell than what maybe a normal body cell would have. Haploid cells only have one chromosome for each of the positions in our karyotype, instead of the normal like body cell that would have two. Okay, so a haploid cell in a human would have 23 chromosomes, or basically you could look at it as one set from one of your parents. That's kind of a biological white lie, because if I say one set from one of your parents, that implies the other parent was useless in making that set, and that's not true. Like, it just means you have 23 chromosomes instead of 46. Some of those 23 chromosomes could be mom's original chromosomes, and some could be dad's original chromosomes, but the key is you only have 23 and one at each position in our actual karyotype. Now, here again is some examples of haploid and diploid cell numbers, just to throw out a reference. And you can kind of see there's garden peas and tomato plants and apple trees, stuff like that on this table. But if we just want to focus on humans, the idea is that humans have 46 chromosomes in their diploid cells, and they have 23 chromosomes in their haploid cells. This fact, as it says here, supports a conclusion written by a very famous guy by the name of Gregor Mendel. Mendel is considered the father of genetics, and we're going to spend a lot of time talking about him in latter parts of this chapter as we're learning genetics. But for now, we just need to understand that Mendel's the first guy to come to the conclusion that, like all offspring inherit one factor, from each parent for everything that we see in the mirror. Later, we called those factors chromosomes, or to be truly accurate, homologous chromosomes. One homologous chromosome, or one homolog, from each parent, okay? So as you see here, Mendel's conclusion is that parental organisms give one allele, okay, ultimately a gene, for each trait to each of their offspring. Now, why did I just use the word allele with the word gene? Well, remember, genes are these tiny little packets of DNA that are loaded on our chromosomes and passed from one generation to the next. And those chromosomes come in matched sets from our parents. And so that means dad gives you like chromosome number 1, mom gives you chromosome number 1, and on chromosome number 1 from each parent, there are genes that are supposed to match up to give you what you see in the mirror, like, I don't know, your hair color, okay? So the idea is to be homologous, chromosome number 1 from mom and chromosome number 1 from dad both have to have a gene in the exact same spot for hair color. Now, exactly what your hair color is from mom and dad could vary a little bit. And if it does vary, then we use the word allele to describe that variation of a gene. So if mom had black hair and dad had blonde hair, right, they're both going to give you a gene for hair color, okay? However, mom's gene for hair color will be black, and dad's gene for hair color will be blonde. So that case would make us use the word allele. Both parents are going to give you an allele for every trait you have through these homologous chromosomes, okay? And as we go through meiotic division, those alleles are separated into separate sex cells. And as they're separated into separate sex cells, then that gives us, natural variation. Whichever sex cells come together as a result of sexual reproduction will determine what the offspring looks like, which is genetics. And we'll learn more about that in a different lecture. Okay, so moving on. Here's a quick little ditty about the idea of homologous chromosomes. Okay, 2 chromosomes of each pair in a diploid cell are called homologous chromosomes, and each of the homologous chromosomes carries genes for the same trait. So let me show you in pictures what I was trying to describe for you a moment ago on that table. Here is a picture of 2 chromosomes, right? These chromosomes basically are from a pea plant, and they're homologous because if you look really closely, you can see that there are A's right here across from each other, D's across from each other, and T's across from each other. So those are genes for the same traits in the same place. One of these chromosomes came from mommy plant, and one of these chromosomes came from daddy plant, but they're carrying genes for the same traits in the same place. Now, you might look at this and go, but wait, Mr. Kennedy, that's a capital A, that's a lowercase a. Those aren't the same. Yes, they are. Okay, because what you're looking at here is not just the gene A. Like if you look close, okay, you've got these plants and they're doing axial versus terminal flowers. Over here, we're doing inflated versus constricted pods, or down here, we're doing tall plant versus short plant. So a lot of times kids want to go, oh, well, that's totally different. Like tall and short, that's not the same. Axial terminal flowers, that's not the same. Well, you're missing the point. Okay, the gene here is not a gene just for axial, it's flower position. So gene for the exact same trait in the exact same place means that these A's are genes for flower position. Those genes are not exactly the same, but they both describe flower position. So that's where we use the word allele. Okay, like axial is a flower position, terminal is a flower position, tall and short, those are genes for height, right? So big T for tall, little t for short, that's genes for the same traits in the same place. They're slightly different from one another, so we refer to them as an allele. Now, once we get into genetics, you're going to start to understand which of those 2 alleles we're going to see in the mirror. That's the rule of dominance and recessiveness, and a discussion for a different day. Now, let's get into meiosis. Okay, so here's the thing. We've already kind of talked about the fact that in order to sexually reproduce, you have to take 2 cells from 2 separate individuals and combine them, right? Well, remember, mitosis produces copies of diploid cells. We can't take 46 chromosomes from a body cell in a human and combine it with another 46 chromosomes in a body cell from another human, right? Because we'll get a baby that has 92. That's why we have to have meiosis. Meiosis is what's referred to as reduction division. It reduces the total number of chromosomes in a cell by 1/2, okay? So if we're thinking about some practical examples, here's the idea with the pea plants that were mentioned in that previous slide. If pea plants have 28 chromosomes and 2 cells from 2 separate pea plants come together, the offspring are going to have 56. Not good. So that's where meiosis comes into play. It reduces the number of chromosomes by 1/2. And the cell that it produces, there's a new term on this slide. It's this G word, okay? Gametes. Gametes is a fancy word for sex cells. So all sexually reproducing species produce sex cells through meiotic division, and we call them gametes. When 2 gametes come together to make a fertilized egg, well, we call that a zygote, which will eventually develop into, well, who knows what? Cat, dog, fish, frog, a plant, a person, whatever, okay? Let's look at how the process works, okay? So in terms of meiosis, just like mitosis, there are phases that you got to know, and you're going to have to be able to recognize them both in words as well as in pictures. So If I go through it too fast in this video, remember, you got a pause button over there on your side of the screen. Use it. Make sure you get this. So we're going to go through these phases. They should look fairly similar, at least in terminology, to mitosis, but there are some subtle differences in what is occurring during each phase. Let's begin, okay? The first phase of meiosis is interphase. Now, bear in mind, I'm ignoring the rest of the cell cycle that could be going on, like outside of, of this. I'm just going to focus on our depth and detail on the physical steps of meiosis. So interphase that like makes up the majority of the cell cycle, this is all I'm going to tell you about interphase. Basically, the cell replicates its chromosomes and hangs out here for a while in preparation for meiotic division. In mitosis, I gave you a lot more detail. After replication, here's the interesting thing. Your cell wants to reduce its genetic material by 1/2, but before it goes through meiosis, it's actually going to make an extra copy of its genetic material. And people go, what? Like, why? Why make a copy if I want to have less? And why make more? Well, because you're going to go through 2 phases of division. And like, we need to have cells at the end that have half the chromosomes we started with, not 1/4. So we got to beef it up a little bit and then go divide twice so that we can eventually arrive at that 1/2 number. I don't know. It's math. It's fractions. You'll see what I mean as I go. Okay? So let's start in prophase. Prophase, first thing I want to throw at you, has a 1 after it here, and that's because there's 2 separate division cycles that occur in meiosis. So there's going to be prophase, metaphase, anaphase, and telophase, all with 1s after them, because it's going to happen like twice. So there's going to be a prophase 1, metaphase 1, anaphase 1, telophase 1, and then guess what? There's going to be a prophase 2, metaphase 2, anaphase 2, and telophase 2. Because it happens twice. In prophase 1, a lot of what you saw in mitosis happens here, but there's a new little interesting twist. So the chromosomes coil up, spindles form. As the chromosomes coil, the homologous chromosomes line up with each other gene by gene along their length, and they form a quartet, if you will, that we call a tetrad. Now, here's the crazy thing that happens in meiosis that did not happen in mitosis. It's called chiasmata, or crossing over. The chromatids in the tetrad pair so tightly that pieces of non-sister chromatids can actually break off and trade places. That's what crossing over is all about. And that's super cool because that gives you variation in offspring that is outside of what you see in either of your parents. Here's what I mean. Like if mom has brown hair and brown eyes and dad has blonde hair and blue eyes, you would expect the children to all look like one parent or the other. They're either going to have brown hair and brown eyes or blonde hair and blue eyes. Well, what happens if a kid is born with brown hair and blue eyes? Neither parent has that combination. How did it happen? That is how it happened. That crossing over. Like you could get a gene like move from one sister chromatid to another and create a new combination of traits that mom and dad didn't have. That's called genetic variation, and that is literally the #1 benefit for sexual reproduction in the animal world, in the plant world, in the world period, right? Because if we were all clones, if something made one of us sick, it would make all of us sick. But because of genetic variation, we have some natural resiliency and immunity to a wide variety of things because we're not just like an identical clone of our parents. So this is huge. Genetic variation. Wow. All right. Now, crossing over can occur at any location on a chromosome, and it can occur at several locations at the same time. So it's not just limited to like one gene at one time. This is what it looks like in a picture sense. So we've got our tetrad here, and if you look close, we've got the legs of non-sister chromatids that are touching, and look what happens down here. Okay, we've got new combination of traits that did not exist in the parents. Now, depending upon which one of these successfully fertilizes a gamete from a mate, Well, that'll determine what the offspring end up looking like as genes are expressed through the idea of dominance and recessiveness, which we'll learn about more in another video with our buddy Gregor Mendel. All right, so crossing over results in new combinations of alleles on a chromosome. Enough said about that. Let's go to the next phase, Metaphase I. This should look really familiar. Same as mitosis with just a tiny tweak. So the chromosomes are going to line up on the metaphase plate or the equator of the cell. This time, though, they're lining up side by side horizontally, okay? Not vertically single file. That's the difference. So watch what happens when this cell divides now as it goes into Anaphase I. What? The chromosomes are pulled to opposite ends of the cell. The centromeres did not break. Sister chromatids were not pulled apart. We literally pulled whole chromosomes to opposite ends of the cell. We've basically reduced our chromosome count by half in the cell. If we look at this cell and this cell specifically in the previous slide, right, and I'll go back, there's 4 chromosomes in there, right? When anaphase happens, there's 2. So we cut the number in half. You might think, well, we're done in meiosis now, right? No, we're not done, because remember, each of those chromosomes that we've separated still has a sister chromatid attached, and that's what meiosis II is for. Breaking the centromere, separating our sister chromatids. Eventually, we're going to produce 4 cells in this meiotic division. All right, so anaphase, the homologous chromosomes are pulled to opposite ends of the cell. This is our reduction division. Telophase I, opposite of prophase I, basically the cell starts to divide in half, right? That's the cytokinesis piece. The nuclear membrane might reform, and the cell can kind of pause here until meiosis II is needed to produce a gamete. As soon as meiosis II is needed to produce a gamete, well, we're going to get a second series or a second set of divisions that literally look identical to mitosis. There's a prophase II, metaphase II, anaphase II, and telophase II to finish off the division of this cell. Okay, so I'll run you through them pretty quickly. This is what prophase II looks like. Look familiar? It's literally just like mitosis. So the nuclear membrane is going to break down. The chromosomes are visible, right? They have the pretty little X structure already. There is no interphase between meiosis I and meiosis II, so we're not duplicating again. The chromosomes are already duplicated. That's what the X structure is for, right? So basically, all we're trying to do here is separate that X into 2 pieces or get the sister chromatids apart. Okay, these cells only have 23 chromosomes in them if we're talking about humans, but there's 46 sister chromatids if you want to count it like that. All right, so that's prophase II. Here is the next phase. This is metaphase II. Look what's up. The chromosomes are lined up single file down the center, again, just like mitosis. They attach to the spindle fibers, and when the spindle fibers contract, whoop, there it is. Okay, the centromeres break, sister chromatids are migrating to opposite ends of the cell, and cytokinesis kicks in and separates what was once 2 cells into 4 total cells. Each of those cells now has 23 chromosomes or 23 sister chromatids, depending on how you want to look at it. Those cells will still have to go through a maturation process in order for them to be a functional gamete, but dude, this is how gametes are formed. So these are our sex cells. So at the end, right, of meiosis II, we have 4 haploid cells that are formed from 1 diploid cell, and those haploid cells will become gametes that transmit our genes to our offspring, 1 generation to the next. Any sexually reproducing organism does this. All right. Last thing I'm going to remind you of is this is huge for genetic variation. All right. The number 1 reason why meiosis occurs, sexual reproduction occurs, again, is so that we are not identical to our parents. Anything that sexually reproduces needs that genetic variation to increase the likelihood of survival from one generation to the next. All right, ladies and gents, that is meiotic division compared to mitosis from chapter 11 of your book. I hope you've learned a little bit. No, I hope you've learned a lot about cell division. I'm Mr. Kennedy, and I will see you next time.